Welcome back. Today we're starting Chapter 14, Antimicrobial Drugs. This is one of the most clinically relevant topics in microbiology. In this chapter, we'll discuss how antimicrobial drugs work, why microbes develop resistance, and what health professionals can do to encourage responsible use of antimicrobials. One of the most famous antimicrobial drugs out there is penicillin, and it was first mass-produced in the 1940s. It saved millions of lives during World War II, and it was truly revolutionary. But the same drug that once ended these epidemics is now part of a new crisis, antibiotic resistance, and this is largely due to overuse and misuse of antibiotics. We'll start with 14.1, and here we'll learn how to compare natural, semisynthetic, and synthetic drugs, recognize early uses of antimicrobial chemicals, and identify key individuals and milestones in antimicrobial development. Most people associate the term chemotherapy with treatments for cancer, but chemotherapy is actually a broader term, and it really refers to any use of chemicals or drugs to treat disease. Antimicrobial drugs are chemicals that target microbial structures or enzymes to kill or inhibit their growth, and this is ideally without harming the host. Even ancient societies practiced a form of chemotherapy. In Nubia, beer-containing Streptomyces bacteria, this is a tetracycline-producing bacterium, was used to treat wounds and gum disease. Chinese and Indian herbalists used plant extracts with antimicrobial properties, and in the 70s, Tu Youyou revisited an ancient Chinese text and isolated artemisinin from the plant Artemisia annua, and this became a cornerstone treatment for malaria. And cultures around the world used moldy bread or fungi on wounds, really unknowingly applying the same principle that led to modern antibiotics. This checkpoint asks us to give examples of how antimicrobials were used in ancient societies. So we talked about the Nubian beer, the Chinese and Indian herbal medicine, Tu Youyou's discovery of artemisinin, moldy bread and fungi. So these examples really show a purposeful use of these antimicrobial substances before modern drug discovery. So what these examples have in common is they show that long before we even knew the term or used the term antibiotic, humans were already kind of harnessing natural resources to fight infections. In the early 1900s, German physician and scientist Paul Ehrlich searched for what he called a magic bullet. These were chemicals that could kill pathogens without harming the human, without harming the patient. And in 1909, his assistant, Sahachiro Hata, discovered salvarsan, or also known as compound 606, which successfully treated syphilis caused by the bacterium Treponema pallidum. And this was the first systematic drug screening approach and a major step toward rational drug design. So this was an approach where they systematically screened a wide variety of compounds, and this remains a common strategy for the discovery of new antimicrobial agents even today. In the 1930s, Josef Klarer, Fritz Meitz, and Gerhard Domack discovered Prontosil. This is a synthetic dye that was found to cure bacterial infections, and its active component, sulfanilamide, became the first synthetic antimicrobial drug. These are man-made compounds that are not found in nature. So this discovery launched the era of a group of drugs called sulfa drugs and earned Domack the 1939 Nobel Prize. A few years before that discovery of Prontosil, Alexander Fleming noticed a contaminating mold, which I can see here and here, called Penicillium notatum that inhibited Staphylococcus growth. So here I can see some Staphylococci bacteria here as well. And you can see the region between the mold and the bacterium. There is a clear region where there are few or no bacterial colonies. So Penicillium inhibited Staphylococcus growth. And when they examined that area more carefully, they discovered penicillin. So this was the first natural antibiotic found in nature, and it was effective against a variety of bacteria and still is, including Streptococcus, Meningococcus, and Corynebacterium diphtheriae. Just be careful when you're looking at the names. So penicillium is the mold, and the antibiotic it produces is penicillin. Then in the 1940s, Howard Florey and Ernest Chain purified and mass-produced penicillin, and they had great success in demonstrating that penicillin was very effective in both animals and humans. Along with Alexander Fleming, they received the 1945 Nobel Prize, and later Dorothy Hodgkin determined penicillin's structure, and this really allowed chemists to develop semisynthetic penicillins that had improved stability and broader activity. So semisynthetic drugs are chemically modified derivatives of a natural drug. And these chemical modifications could do the following. For instance, increase the range of the bacterial cells that are killed, increase the stability of the drug, decrease the toxicity of the drug, or give additional beneficial traits for treating infections. So together we have Alexander Fleming with his accidental discovery of penicillin, Florey and Chain over here mass-producing penicillin, and Dorothy Hodgkin, her structural work, all together mark the birth of the antibiotic era. Also in the 1940s, Selman Waksman at Rutgers University studied soil microbes and discovered actinomycin, streptomycin, and neomycin from the bacterium Streptomyces. And Streptomyces is part of a group of bacteria known as actinomycetes. These produce more than half of all known natural antibiotics. So this is a good reminder that soil is a vast reservoir of antimicrobial compounds and resistance genes. Our next checkpoint asks, why is the soil a reservoir for antimicrobial resistance genes? So soil microbes, especially the group Actinobacteria, such as Streptomyces, they naturally produce many antibiotics to compete with neighboring microbes. But because of this constant competition and kind of chemical warfare of their antibiotics, soil microorganisms have evolved and they have resistance genes, which they sometimes share to protect themselves from these natural antibiotics. So the soil contains both antimicrobial producing organisms, but also microbes carrying resistance genes and resistance mechanisms, so they serve as a reservoir of resistance genes for potential transfer to pathogens. All right, let's recap 14.1. The first learning objective asks, compare and contrast natural, semisynthetic, and synthetic antimicrobial drugs. So natural antimicrobials, these are produced directly by living organisms. For instance, the penicillium mold that Alexander Fleming discovered contaminating his plates, we found that produces the natural antibiotic penicillin. So these are this is discovered in nature, and they're not chemically modified. For semi-synthetic antimicrobials, these are chemically modified derivatives of natural antibiotics, and they're created to either expand the spectrum, like what can be killed, improve stability, or reduce toxicity. So we have examples of modified penicillin that were developed after its structure was determined by Dorothy Hodgkins. And finally, synthetic antimicrobials. These are fully man-made chemical compounds. They're not found in nature, and an example is sulfanilamide from the dye Prontosil. That was the first synthetic antimicrobial. For the second learning objective, describe the chemotherapeutic approaches of ancient societies. So we saw that ancient societies or cultures unknowingly used natural antimicrobial compounds. The Nubians brewed Streptomyces containing beer, and they unknowingly had tetracycline exposure. Chinese and Indian herbalists used plant-based medicines with antimicrobial properties. And then we saw also moldy bread or fungi were used to apply to wounds for healing. So these examples really represent early empirical chemotherapy using these natural substances to combat infection long before what we know today. long before modern drug discovery. For the third learning objective, describe the historically important individuals and events that led to the development of antimicrobial drugs. We had Paul Ehrlich in 1909. He developed salvarsan, or compound 606. This was to treat syphilis. This was the first targeted chemotherapeutic agent. Remember, these were the magic bullets used to kill bacteria without harming the human. We had Gerard Domack in the 1930s, discovered Prontosil, the precursor to sulfanilamide, which was the first synthetic antimicrobial. We had Alexander Fleming in 1928, who accidentally discovered penicillin, the first natural antibiotic, from his moldy plates that were contaminated with penicillium mold. We had Howard Florey and Ernest Chain in the 1940s, who purified and mass-produced penicillin and showed its effective use in clinical settings. Dorothy Hodgkins in 1948, Hodgkin, not Hodgkins. It's Dorothy Hodgkin determined penicillin structure, enabling the creation of those semisynthetic penicillins. And Selman Waksman in the 1940s discovered streptomycin, actinomycin, neomycin from that soil bacterium Streptomyces. He also coined that term antibiotic. The results were to lay the foundations for modern antimicrobial theory and drug discovery screening methods. So far, we've seen how antimicrobial therapy evolved from ancient herbal remedies to synthetic compounds and natural products. Next, in this section, 14.2, we'll focus on the principles of antimicrobial chemotherapy. This is how we classify, dose, and combine these drugs in clinical practice. When choosing an antimicrobial drug, clinicians have to consider, one, the mechanism of action. So do we need something that's, for instance, bacteriostatic or bactericidal? The spectrum of activity. So do we need something that kills many different types of microbes, maybe gram-positive bacteria, gram-negative bacteria, and fungi? Or do we want something that has a narrow spectrum and just, for instance, kills gram-positive bacteria only? We also need to look at the dosage and route of administration, as well as side effects or drug interactions. Let's do a quick refresher on those suffixes. So bacteriostatic drugs, remember, these are drugs that inhibit the bacterial growth temporarily. If you were to remove the drug, then the cells would grow again. And we use bacteriostatic drugs when we want to lower toxicity, and we assume that the human immune system can clear off the rest of the microbe. In contrast, bactericidal drugs will kill bacteria outright, and the choice again really depends on the patient. We use bactericidal drugs for immunocompromised patients or for severe infections, such as endocarditis. In regard to spectrum of activity, we have narrow spectrum drugs that target specific bacteria. For example, they might only target gram-positive bacteria, whereas broad spectrum, these drugs cover both gram-positive and gram-negative organisms, and they're useful when there's an infection by unknown pathogens, you're not sure what's causing the infection, or mixed infections, maybe multiple species are causing the infection. The problem with broad spectrum drugs is that they could lead to superinfections, which are secondary infections that occur during or after the pre-existing infection. And we see this in yeast infections such as candidiasis or pseudomembranous colitis caused by Clostridioides difficile, which can be fatal. And here we can see kind of an example of how this happens. If you're in normal cases, we have a lot of different normal microbiota, which could keep a pathogen in check, right? The normal microbes are using up nutrients and space, so the pathogen can't overdivide, right? Overgrow. But if we're taking broad spectrum antibiotics, it'll kill off everything that is not resistant to the drug, and the pathogen remains alive. So these drug resistant pathogens create a problem because now they can proliferate, they can overgrow, causing that superinfection. This checkpoint asks, what is a superinfection and how does one arise? So a superinfection is a secondary infection that develops while a patient is being treated for a pre-existing infection. And it often happens when a broad-spectrum antimicrobial drug kills the normal microbiota, so that's normally protecting us. And this allows resistant organisms to overgrow. So an example could be Clostridioides difficile overgrowth, because they make those endospores, and this can lead to a life-threatening condition called pseudomembranous colitis. Another example is for yeast infections caused by Candida. These can occur after antibiotic use. So for instance, in the body, such as in the vagina, we have both bacteria and the yeast, Candida, but if you take antibiotics and kill off the bacteria, the yeast can overgrow. We also have to consider dosage. So dosage is the amount of drug given at a certain time interval. And we aim for therapeutic levels that are high enough to inhibit the pathogens, but without harming the patient. So factors include, for instance, body mass and age. For example, in children, the dose is based on their body mass, whereas in adults, we generally have a standard dose. Organ function is also important. So the liver and kidneys usually help us clear the drug. And if there is some kind of, if there are problems with the liver or kidney, then that means there will be slower clearance of the drug, which can lead to toxicity or greater toxicity. Half-life of the drug is also important. Half-life tells us the time it takes for half of the drug to be eliminated from the blood or plasma. And a shorter half-life would mean more frequent dosing of their drug is needed, whereas a long half-life for the drug would mean that we don't need as frequent of a dose, but there's greater risk for toxicity. Whether the drug is dose-dependent or time-dependent is also important. Some drugs are dose-dependent, and this means that they're more effective when administered in large doses in order to provide high levels of the drug for a short time at that site of infection. Whereas other drugs are time-dependent. This means that they're more effective when lower levels are maintained over a longer period of time. The route of administration is also important, and in general, or the trend is that the more invasive the route of administration, the greater the dose you'll receive in a shorter period of time. But the easiest route of administration is often the oral route, right? You're taking a pill by mouth, for instance. This is convenient and it's easy to do at home, but the drug might not always be very well absorbed through this route. Topical agents are good for skin infections that are localized, for instance, applying bacitracin or different antifungals on the surface of the skin. We also have parenteral routes, such as intravenous or intramuscular. These deliver drugs directly into the bloodstream for rapid, high concentrations, which is often necessary for severe infections. In this graph, we can see on the y-axis the plasma concentration of the drug, and on the x-axis we have time, where time 0 is the time at which the drug dose was administered. So we can see that IV, the red line, IV administration achieves the highest plasma levels immediately, whereas the oral in blue and intramuscular in green, these routes rise more slowly but can still reach therapeutic ranges. This checkpoint asks for a list of 5 factors to consider when determining the dosage of a drug. So first we have to think about patient characteristics, right? What is their age? If they're a child, we have to consider their body mass, and we also have to consider their immune system, their immune function. For the second one, organ function, we have to think about their liver and kidney health, because this can affect the metabolism and excretion, getting rid of the drug. The drug half-life is also important. How long does it take to clear the drug? So remember, half-life is how long it takes for 50% of the drug to be cleared from the blood, from the plasma. The drug type is important, whether it's dose-dependent where you need a high concentration, or time dependent, where you need a lower concentration at steady levels for a longer period of time. And also route of administration. Is it going to be oral, topical, or parenteral, which means IV or IM? These routes can influence the absorption of the drug and the plasma concentration of the drug. For the second one, name some typical side effects associated with drugs and identify some factors that might contribute to these side effects. So typical side effects include things like allergic reactions, upsetting the gastrointestinal system, liver or kidney toxicity, and potentially secondary infections. So this can be due to high or prolonged drug levels, especially if the drug has a long half-life. That can trigger allergic reactions or GI problems or toxicity. This could be poor drug clearance due to liver or kidney dysfunction, or interactions with other drugs that the patient's taking at the same time, and that these other drugs or combination of drugs can alter metabolism of the drug as well as elimination of the drug. In terms of drug interaction, drugs can interact in two major ways. So we either have a synergistic effect. This is when the combined effect of the drugs is greater than their individual effects. So an example from our book talks about trimethoprim and sulfa methoxazole. These are individually bacteriostatic, but if you combine the two, they're bactericidal. Another example is this picture that I'm showing you up here. I have, let's say, antibiotic 1, which has this moderate zone of inhibition. Another antibiotic with a second, pretty moderate zone of inhibition. But when I combine the two, you can see a much greater zone of inhibition. So that would be a synergistic effect. The other type of interaction is antagonistic. This is when one drug reduces the effect of another drug. For example, antacids might decrease the absorption of certain antibiotics that require high acidity to be absorbed. And in our book, they also mention something that is, unfortunately, does happen in some patients, where some antibiotics can reduce the effectiveness of oral contraceptives. So this checkpoint asks us to explain the difference between synergistic and antagonistic drug interactions. We saw synergistic interactions are when two drugs work better together than they do alone. So we saw the example of trimethoprim and sulfamethoxazole. That one's actually sold as Bactrim. These are bactericidal when we combine the two. Alone, they're bacteriostatic. For antagonistic drug interactions, this is when one drug will reduce or block the effect of another. For instance, antacids raise the pH of our stomach, and that might reduce the absorption of acid-dependent antibiotics. Toward the end of section 14.2, they talk about ethics and resistance regulation. So there are two main human behaviors that are driving antimicrobial resistance, where the microbes no longer respond to the drugs. So one is stopping the treatment early. A lot of patients, for instance, will take antibiotics, and they're supposed to take a dose maybe for 10 days, but then they feel better after maybe 5 days, and they don't finish the full course of drugs, leaving resistant bacteria behind to spread later on. So treatment for tuberculosis is a good example, because the recommended treatment for tuberculosis lasts from 6 months to a year. So imagine you have to take medications for this long period of time. Unfortunately, most individuals with TB live in underdeveloped or underserved regions where we have lower adherence rates. The second problem is overprescription of antibiotics. For instance, prescribing antibiotics for viral infections, which do not do anything, right? Antibiotics kill bacteria, not viruses. So solutions to these issues include better education or perhaps directly observed therapy, especially for long-term treatments like tuberculosis. So indirectly observed therapy, this would be the supervised administration of medications to patients. So patients would have to come into the healthcare facility to get their medication, or healthcare providers would go out to the community and administer the medication to the patient, either in their homes or another designated location. And our book also shares results from a study that shows a rise in parents expecting antibiotics for their children, especially in lower income groups that are shown in blue. You can see the blue line is steeper than the red. This really highlights the need for public education about responsible antibiotic use, as well as what antibiotics can actually treat, right? They can treat bacterial infections, but not viral infections. So I think the survey questions are also very interesting. In this one, for instance, they say the parents would rather give their child an antibiotic that might not be needed rather than wait and see if the child gets better without it. So I can understand the frustration on the parents' end, right? Your child is sick and you're going to the doctor and the doctor's not giving you any medication to treat, right? Treat your child. So unfortunately, a lot of these viral infections, you just have to wait it out. Maybe you can lower the fever with medication, but antibiotics would not do anything for that. This is a nice image that shows you what would happen if you were to stop a dosage of antibiotics early. Let's say that you're supposed to take ciprofloxacin for 7 days after a urinary tract infection, but the patient feels better after 3 days. So they feel better. They're like, oh, maybe I should just save these meds in case I get sick again in the future. So this is on petri dishes, but you would see a similar effect in the body. So let's say that this population of bacteria are causing the urinary tract infection. Most of the bacteria are sensitive to the antibiotic, but some of them are going to be resistant. So if you take the antibiotic, let's say the patient's taking it for the 3 out of 7 days, you're going to kill off the sensitive species, but the mutants that are resistant to the antibiotic will not be killed. And then it doesn't kill off all of them because the patient stopped early, right? Taking the drug for 3 days instead of 7 days. What happens when you put the drug away, you're saving it for the future, is now those resistant strains grow, right? They start growing. And then eventually they overgrow, the patient feels sick again, they take the same drug, but it's not going to be effective because most of the cells are now resistant. And remember, through conjugation and other types of transfer of genes horizontally, remember horizontal gene transfer, the resistance can spread as well. Now, if the patient did take the full dose for 7 days, what would have happened? So if they took the full dose of antibiotics for the 7 days, you would have killed off all those sensitive strain, and the additional days of drug would also help and work with your immune system to also kill off the resistant strains in addition to any remaining bacteria here. So this would have helped us avoid this future situation. All right, let's recap 14.2. The first one is contrast bacteriostatic versus bactericidal antibacterial activities. Bacteriostatic drugs will inhibit bacterial growth and reproduction, but this is reversible. So the cells will grow again if you take away the drug. This relies on the host immune system to eliminate the remaining microbes. Whereas in bactericidal drugs, this kills bacteria directly, and it's necessary for patients who are immunocompromised or have life-threatening infections. For the second one, contrast broad-spectrum drugs versus narrow-spectrum. So narrow-spectrum drugs target specific bacterial groups, for instance, only gram-negative, and this minimizes the disruption to normal microbiota. Whereas broad-spectrum drugs treat a wide variety of bacteria, for instance, killing both gram-positive and gram-negative, and we use these when the pathogen is unknown or for mixed infections. The problem with these is they can disrupt our normal microbiota, our normal flora, and lead to superinfections. For the third one, explain the significance of superinfections. So superinfections are secondary infections that occur during treatment for another infection or right after. It's caused when broad-spectrum antibiotics kill off our good bacteria, right? Eliminate these protective microbiota and allow the resistant organisms to thrive. The fourth one, discuss the significance of dosage and the route of administration. So dosage is the drug concentration at the infection site, and we want to minimize toxicity. It's influenced by patient mass, their organ function, the drug half-life, and whether the drug is time-dependent or dose-dependent. The route of administration can be oral, that's convenient, but can be poorly absorbed. Topical is for localized skin infections. Parenteral, which is IV or IM, these are rapid. You're going to get quickly a high dose of the drug into the blood, but it's invasive, right? These are used in severe infections or when the oral route's not possible. For the fifth item, identify factors and variables that can influence the side effects of a drug. So this could be drug-related factors like the half-life, the dosage, and the metabolic pathway of the drug. It could be patient factors, like if they have a liver or kidney impairment, they have slower clearance of the drug. Drug interactions are important. And so we have to think about toxicity, allergic reactions, or secondary infections. For the last one, describe the significance of positive and negative interactions between drugs. So positive are the synergistic interactions where two drugs together have a greater effect than when the drugs are used alone. Whereas negative antagonistic interactions where one drug reduces the activity of another drug, or it could even increase the toxicity of the other drug. And that wraps up part 1. So we've traced the origins of antimicrobial therapy and reviewed the core principles of drug selection and administration. In the next video, part 14.3, we'll dive into the mechanisms of how antibacterial drugs work at the molecular level.